I would like to present our results of microbial diversity analysis across the Atlantic Ocean, where we used an integrative approach to assess patterns of microbial diversity in the context of productivity and their hydrographic environment. Marine microbes play a key role in marine ecosystems. They form the base of the marine food web by fixing inorganic carbon through primary production and thus make it available for other organisms. Most of the carbon is recycled in the so-called microbial loop, where bacteria and microeukaryotes access and remineralize carbon and nutrients and thus make the nutrients again available for phytoplankton. The way how these organisms are distributed in the ocean is of course everything but uniform. A concept to describe the community composition across space and time is the assessment of their beta diversity. This means that a composition of communities at each site is compared to other sites in regards to reoccurrence and extinction of the same species, but also changes of the relative abundance of individual species. Due to their fast generation time, microbes are ideal bioindicators to monitor ecosystem stability, the invasion of alien species, pollution, and changes in ocean productivity. Microbial diversity has been traditionally thought to be mainly driven by temperature, but recently this has been advanced in a more holistic and in an oceanographic context, which are so-called the microbial fingerprints, reflecting all the biotic and abiotic environmental conditions. Therefore, microbial samples largely group into ocean basins, as shown by the Tara Ocean project, for example. The microbial communities are here reflected by color, and you can see that they actually follow the ocean basins. In a more refined study by Bolaños et al., they used mean sea surface height to first identify the environmental regions, and then use the cluster analysis of the microbial community to see how it is reflected by the environment. And you can see color-coded that it actually fits well into the analysis of mean sea surface height, but they also identified an outlier which they could link to an eddy which was actually originating in the subtropical region, and thus the microbial community was more similar to these samples than to the environment or the neighboring samples. This shows that the microbial community also persists longer sometimes and then reflects an ecosystem patch in a larger ocean region. So in our study, we analyzed the different ocean provinces and how the beta diversity is linked to productivity. To link the biomass turnover of microbial communities to their pelagic advection, we calculated a length scale for biological physical coupling, a so-called productivity-specific length scale, using a quantitative comparison of measured biomass turnover and measured current speed. So we were basically testing how well a sample represents its environment. We sampled the Atlantic Ocean from south to north, roughly at every half-degree latitude, crossing multiple ocean regions, which were strongly different in their biogeochemical and physical profiles. We adapted the naming conventions and also the definitions by Longhurst, who used physical parameters, so temperature and salinity, and biological, so satellite observations of chlorophyll A concentrations to define ocean provinces. These are of course very coarse-grained, but we're actually pioneering this idea of biological physical coupling, and he noted himself that the boundaries cannot be static and needs to be refined over time. Thus, we used a combination of in situ measurements and satellite data to refine the ecosystem boundaries in our dataset. We used stable isotope incubations of a 13C to measure primary productivity in our microbial communities. Then we used multivariate analysis of metabarcoding DNA analysis of microbial communities to identify patterns in microbial beta diversity across multiple trophic levels. We found that the beta diversity signals clearly reflected the Longhurst provinces in our multivariate RDA analysis, as shown here for the autotrophic eukaryotes. We could also show that across trophic level, these patterns remained or were persistent within the cyanobacteria, the eukaryotic mixotrophs, the heterotrophic eukaryotes, and the heterotrophic prokaryotes. In my talk today, I will only focus on the autotrophic eukaryotes, discussing the more general patterns we observed across all trophic levels. Further, we could identify that microbial beta diversity clustered according to ocean currents. For example, the South Atlantic Gyre province, which Longhurst roughly defined between -5 and -35 degrees south, we identified two provinces which were markedly separated into a more warmer, heterogeneous northern part and a dynamic and colder southern part, separated by the Rio Grande rise and a part of the Brazilian current. This was actually also reflected in our microbial beta diversity, which followed these patterns. And on a more functional level, we found that microbial beta diversity separated into provinces with high and low chlorophyll A concentrations along the first RDA axis across all our groups. So this takes us back to Longhurst, why even though his definition were rather coarse and only on few parameters, it reflects well microbial communities, as it shows that productivity is on a larger scale a major structuring factor of microbial communities. Interestingly, the province with the highest productivity in our data set, as shown here with our incubation experiments, was also the province with the highest spread in beta diversity. The variability and the primary productivity increase is further discussed in the paper from von Appen et al. Using a towed vehicle measuring physical and biogeochemical variables across this transect. So the spread in the beta diversity raises the question, at what scale do the regional biodiversity patterns collapse into local variabilities, or are we just sampling an ecosystem patch? From the perspective of a microbe in the ocean, you are constantly advected by currents. Thus, changes in the environment can act top-down on microbial growth and sustain or does not sustain a species population. On the other hand, due to their fast generation rates, populations can rapidly adapt to new environmental conditions and thus react bottom-up. To take these two components in an ecological context, we calculated productivity-specific length scale for each sample in our dataset. This means that bottom-up we used the specific primary productivity, which is the primary productivity has replaced the standing stock biomass of chlorophyll A concentration, so an approximation of community turnover. And then top-down control, we use the horizontal velocity of the ocean currents measured with ADCP. This results in a mosaic of smaller and larger tiles in a mosaic with short and long productivity specific length scales. So the short productivity specific length scales occur when the turnover is high and the current speed is very low. On the other hand, the beta diversity is expected to only change between sites which are further apart from each other when the current speed is high and the specific primary productivity is low. So looking at our data, if we see that there is a mosaic of different specific length scales, with the highest specific length scales in the southern part and very short, for example, in the middle of the warm south subtropical gyre. This is also shown in the plot where we plotted the specific productivity against the productivity specific length scale. But we also see that there in the middle is a large cloud of a lot of intermediate productivity specific length scales. So to come back on the Canary province I mentioned earlier, and zooming in, we found that neighboring sites with relatively similar productivity length scales and site 157 relatively shorter to the neighboring sites in its productivity specific length scale was also an outlier or an ecosystem patch in comparison to the beta diversity gradient, which is more gradual along the second RDA axis in our multivariate analysis. So in conclusion, we show that microbial beta diversity groups into ocean provinces with high and low chlorophyll A provinces more similar to each other. Our calculations of a productivity-specific length scale provide a first-order estimate of how spatially extended a microbial diversity signature may be. We could also show that the length of the scale reflects patchiness in microbial beta diversity. As a future perspective, we could apply Lagrangian observation with overlapping productivity length scales, and then we could resolve ecological processes on a finite resolution and validate our observations of this study. We could then use this to identify ecosystem boundaries to better understand and monitor our ecosystems and the ecosystem functions. Thank you.